So we saw that it's very important to select the strain and also what could be the impact of fermentation parameters. This is the study case. This study case actually want to have for objective to confirm and illustrate the importance of the yeast strain selection, the fermentation temperature, and also the nutrition regime during fermentation. Because the idea is actually will be to select the best strain and optimize fermentation conditions according to the winemaker's target. So the idea is actually to focus on either yeast strains and fermentation parameters and to see the impact on Sauvignon Blanc. So for this, we took a Sauvignon Blanc from the Loire Valley that you have the characteristic here. We fermented in 50 liters tanks to have the possibility to make a tasting at the end, and we adjusted the ratio YAN on sugar to 1 with different nutrients. And these different nutrients were added half of at yeast inoculation and half at third of the fermentation. But what were really so the conditions? So we took two strains, the strain A from previous experiment and the strain 17912, that is actually the one that was the best rated from the previous experiments. So we selected these two strains. Then we applied 3 different fermentation temperature, either 12 degree constant or 3 days at 18 degrees C at the beginning and then in the 12 degrees C room chamber, or 18 degrees C constant, and to see the difference between this temperature regime. But in addition to that, we also selected 3 nutrition regimes. 3 nutrition regimes that were either mineral nitrogen, so DAP, diammonium phosphate at the yeast inoculation, and then a yeast autolysate at third fermentation, or the reverse, or only spring firm extreme, so only a yeast autolysate. So 2 strains, 3 temperature, and 3 nutrition regimes, and we will see the impact. Just to come to make a small focus on the spring firm extreme, what is the spring firm extreme? The spring firm extreme is actually what we call a total yeast autolysate. So briefly and simply, you take yeast, cream, and active yeast, you kill it by a heat treatment. This heat treatment destabilizes the enzymes. You make an inactivated yeast. And with temperature and pH regulation, you make the own enzymes of the yeast active to degrade itself. And this degradation is a real autolysis. And this autolysis can go to the end and make what we call a yeast total autolysate, meaning that you have almost all the intracellular content of the yeast that was solubilized. And this is what we call the spring firm extreme. So it's a source of organic nitrogen, but also other nutrients, lipids, sterols, vitamins, minerals. But in that case, it's a pure organic product. Its content in available nitrogen is actually quite low. If you compare to the diammonium phosphate, in the diammonium phosphate, you have 21% of yeast available nitrogen. In the total yeast autolysate, you have about 3.7%. But you don't have only nitrogen in the spring firm extreme. You also have all the other nutrients. So that's why for this experiment, we considered that the spring firm extreme contained 10% of YAN equivalent according to its fermentative power. So 10% for the spring firm extreme and 21% for the DAP. The results are interesting to see. This is the focus on temperature for timing. You see the temperature at 18 degrees C or 12 degrees C here, or 18 starting and then going to 12 degrees C. And what you see is actually between the strains, you have very similar between strain A and strain 17912. It's very similar. What you see as well is that the fermentation at 12 degrees C here, and the fermentation at 18, then 12 degrees C, approximately finished at the same time. If you focus on the nutrition regime, so this is the 12-degree constant, end of fermentation. This is the 18-12, and this is the 18. What you see is that actually you have more difference at 12 degrees C than at 18, 12, or 18, where the yeast are almost finishing at the same time, whatever the nutrition regime. But at 12 degrees C, what you see is that you finish better with the light color, and the light color is actually in each case for both strains is when you have spring firm extreme at the beginning and spring firm extreme at 1/3 of the fermentation, so only organic nutrition. So it means that even if in these organic nutrients you have less available nitrogen, their fermentative power is really making less stress to the yeast to ferment better. Then you see the volatile acidity, the impact on the volatile on the analytical parameters. So you have here 12 degrees C, 18, 12, and 18. What you see on the volatile acidity for strain A is really important. It's the same or approximately the same for the 17, 9, 12. What you see actually is that as far as you go higher temperature, you decrease the volatile acidity, and actually you increase a little bit the acidity. But it's no real difference between the nutrition regimes. When you go to total SO2, it's very different strain. You see the strain A is producing over 100 ppm of total SO2. Let's say, whatever the conditions, the strain 17912 is much cleaner with 60 ppm of SO2. So it's very different. You have a slight difference as well with spring firm extreme addition. Spring firm extreme addition seems to make less stress, and if you have less stress, you have also actually less SO2 production. Then if you go to the higher alcohols and acetate esters, here I put the isoamyl acetate, and again you have strain A on the left and strain 17912 on the right. What you see, it's very different. But what is interesting is when you start to see the difference between the temperature regime. Because on the isoamyl acetate, you have an obvious impact of the temperature. You have much higher concentration of isoamyl acetate at lower temperature, or when you lower the temperature after the beginning of the fermentation, so you limit the evaporation. If you see the 2-phenylethanol and its acetate, it's interesting to see. Again, very different strain, but in the strain producing a lot of these compounds, actually, as far as you have a higher temperature, you increase the production of 2-phenylethanol, and so you increase as well the production of acetate. And you have a little bit more acetate at 18 than 12. Why? Because you limit the evaporation of this acetate. If you go on the ethyl esters, it's more difficult to see. You have difference between strains, but it's more different to see any difference on the nitrogen regime. But what you can see is that at least on the ethyl octanoate and ethyl decanoate, you have like a trend to produce more or to release more, let's say at higher temperature. And this could be due to the length of the chain, octanoate, 8 carbon, decanoate, 10 carbons, that actually are going better through the membrane with a higher temperature, so a higher permeability. But then even more interesting is the thiols, the impact on the thiols. what you see here, you have the three thiols, 3MH, 3MH acetate, and 4-MMP. So you see very different strain. 4-MMP not released at all by the strain A, but released by the strain 17,912. You have higher release of thiols at higher temperature. We see that very well at 3MH and also the 4-MMP. But what we see as well for the fermentative flavors is the same for the 3MH acetate. The 3MH acetate, actually, even if you produce lower 3MH, you have higher concentration of 3MH acetate at lower temperature because you don't lose it by evaporation. And the main, even better interest is that the huge impact of the fermentation conditions on 4-MMP. You see that at 12 degrees C, you have no release of 4-MMP. But in higher temperature, you have very small or no release of 4-MMP when you have DAP, so ammonium phosphate, at the beginning of the fermentation. So meaning that explaining really the IRC7 expression and the nitrogen catabolite repression. So here in that case, an organic nutrition is very interesting to make more 4-MMP through a yeast that is able to release 4-MMP. So if I summarize that on the possible impact on the sensory profile, I grouped together the compounds with tropical fruit notes, greenness, banana candy, floral, other fruits, and that's the possibility. So for the strain A, if you want an amylic profile, it will be better to ferment at 12 degrees C, first DAP, then spring firm extreme. And from the pre-tasting notes, it was actually really affordable. So it was really confirmed. If you do that with the strain 17,912, for sure, in that case, you will act as well on the greenness, so the 4-MMP release. And this 4-MMP release, or this green predominance compared to other flavors, would be nicer at 18 degrees C with spring firm extreme at the beginning and DAP afterwards. This has to be confirmed for sure by professional tasters. It's only pre-tasting notes, and due to the COVID-19 crisis, we couldn't make the tasting for time being. So about this, the validation of the strain in the field, we saw that the 17,912 strain was really best rated for all the production of Sauvignon Blanc. So we made extensive trials in 6 different countries, 32 trials. And these trials are actually rated afterwards. So you only have in one tank the control, yeast that the winery use, and in the other tank, you have the 17,912. And then at the end of the fermentation, you compare the implantation control, meaning if you recover the yeast at the end of the fermentation. And you see that the 17,912 has a very good implantation rate, so no problem in that. Then I will illustrate and finish by an example of results. So it was again in Sauvignon, on Sauvignon Blanc in the Loire Valley. We fermented 17 degrees C with 20 grams of hectoliters of yeast. And this is what we had at the end of the fermentation. So if you see the kinetics of the 17,912 in blue here, compared to the kinetic of the control, in that case, the control yeast, we had quite a short lag phase and fast fermentation. That was interesting. But as well, what we saw is that in terms of end of fermentation analysis, we had actually slight volatile acidity compared to the control and the total SO2, so total sulfites that were about the same than the control. So in average, faster and clean fermentation profile. In terms of organoleptic profile on this yeast, it was really interesting that compared to the control, what we saw from the aromatic compounds is really illustrated here. We had more white fruits, exotic fruits, but no amylic notes, and higher blackcurrant, but boxwood, so green flavors in case of the 17-9-12. And in the mouth, we had a nice acidity maintenance, and also that brings actually a taste persistence and a better balance. So at the end, we confirmed by field trials that this yeast was particularly interesting, especially for thiolic varieties and especially for Sauvignon Blanc. So as conclusion, briefly, we know that Sauvignon Blanc is mainly driven by fermentation flavors and varietal flavors, the thiols. The selection of yeast, we proved that it's crucial to express this potential, either through the different type of ester that is produced, but also through the IRC7 gene expression. Then after the yeast, actually, after the strain, that is the most important parameter, you have the fermentation temperature. And the fermentation temperature is really impacting the flavors. And what we saw is that when you start at high temperature, then you lower the temperature during the fermentation. It was like a best compromise in terms of release, production, and conservation of aromas, because you produce more aromas at the beginning and you keep more aromas at the end. But you have to know that if you do these kind of things, it's as slow as a low temperature fermentation. So depending on the, let's say, the possibility you have in cellar, it could be also a bit difficult to do it. Then after that, so the yeast strain, after the temperature, you have the effect of the nutrition. The nutrition effect is lower, for sure, but can be drastic. It can be drastic on the, for example, on the release of 4-MMP, we saw that the ammonium supplementation at the beginning was lowering or even blocking this release. And then we saw that the organic nutrient was a little bit lowering the stress of the yeast as well.
Natalia, do not translate. Etienne, juste 4 minutes pour les questions, il faut finir.
So at the end, and to conclude, we had the effect, so the impact on the yeast on Sauvignon Blanc. We saw that the selection of the yeast is drastic, but you have the selection of the yeast, and after the selection of the yeast, you can apply some different conditions of fermentation. First, the temperature, secondly, the nutrition to overexpress the potential of thiol profile of the Sauvignon Blanc. So this strain is now available in market. So the 17-9-12 is now the SAF-NO SH12 in the market. So I would like to thank, let's say to conclude, we'd like to thank our partners in Spain, so Enforum and Amphikimica, that are sponsoring as well this module.